further on the DNA marker, on the STR and VNTR, you know that it's repetitions, and scientists apply it to build what we call the DNA fingerprinting. And DNA fingerprinting was applied to investigate the potency testing. Have a little bit of today. Apply a DNA marker to investigate on potency testing and forensic science. However, we also know that in yesterday, we also know that if a result of the potency testing and also forensic science is false, that means the DNA is not matched. This conclusion is 100% right. There are no doubt about it. But if the conclusion is positive, that means there's a match. This conclusion can never 100% right. It only close to 100%, right? I mean like 99.998%. But it can never obtain 100% right. Because there are always someone in the population who have the same genetic background with the 16 STR marker with the crime scene of the father in case of potency testing. And now we will see also a case that can provide a false negative. I repeat, this case is really rare. So in this case, I took a picture from the figure in Express. You can see there's a man, US citizens, who bring her son to the lab to test for the DNA check, and the result turned back that it's not the father of the child. The DNA is not matched. We know that if the conclusion is wrong, it's 100% wrong. But in this case, the wife is where that she's not cheating, not have a affair or anything. And when scientists is cover further in the genome of the man, they see that in a man have two sets of genomes. So this rare phenomenon is called the human chimera. Chimera is actually a fictional creature in the Greek ancient story. Have a head of the eagle, the body of the lion, tail of the snake. In human chimera is a term of genetics to see that an individual can have different genome sets. So how is it so? Normally, when we have a twin, we have two cases twins, right? The first case is identical twin. Or sometimes we call it homozygote twin, or same zygote twin. The identical twins, it comes from when the egg from mother combined with sperm father to develop into the zygote. This zygote is through mitosis and developing into an embryo, but somehow in development it spread into two different embryos and can result in two different individuals. So in this case, the two individuals will have the same genetic background, right? They look alike. They are have the same genetic background, they have both gender. If one individual is male, that means the other also male. And if one individual is female, the other also female. Because they have the same genetic background, since they develop from the same zygote. Right? Another type of twins is a non-identical twins. Or sometimes we call heterozygote twin because they came from the different zygotes. Is sometimes a woman, instead of having one mature egg in 1 month, in one cycle, can have 2 mature eggs at the same time in one cycle in 1 month. And these 2 mature eggs will combine with 2 different sperm from the father, and in result we have 2 different zygotes. Okay? And these 2 different zygotes is developed into 2 different persons. So in this case, we have a non-identical twins. That means that these 2 individuals will have a different genetic background. They are like normal siblings. They could be different, and they even can have a different gender. Okay, we have some twin whose one individual is male and the other is female. However, in the human chimera, I repeat, this case is really, really rare. Okay? These 2 zygotes somehow combine together during development, combine together and develop into one individual. And this individual, because during the very first cycle of the development, there will be cell that come from one zygote. For example, this one. There would be some cell come from one zygote, and the other cell it comes from another zygote. Leading the consequences is that in here, this individual developed from 2 different zygotes, so inside him there are 2 sets of different DNA. And those, when he taking a DNA test for himself, he will take the DNA from the blood. And the DNA blood comes from one zygote. So it will have a different DNA background. But when he is producing gamete and deliver the gamete to his child, the child will inherit it from the gamete from another zygote. Leading consequences that the DNA of him, or exactly his DNA test from the blood cell, and the DNA test from the child are not matched. But actually, there are no cheating here, no affair here. It's just inside the man himself contains 2 different sets of genomes. Okay? You understand this case, right? But I repeat, it is really, really rare. So please do not use as a pills or anything, because it's too rare. It just actually is the first case in history we have the he will create a timer. So the last DNA marker that I want to introduce to you today is the single nucleotide polymorphism. Like the name, it's a genetic polymorphism. That means it's different between individuals, but it's composed by only one single nucleotide. One single nucleotide differences in 3 billion base pair. So, for example, and remember that SNP or single nucleotide polymorphism is the most common in genetic polymorphism. Actually, 90% of the genetic polymorphisms are SNP. Only 10% is repeatation, microsatellite, TR, and all other stuff. So it differs in only one nucleotide, and if the 2 alleles is different, we call it the heterozygous. And if the 2 are the same, we call it homozygous. For example, the SNP in red here. We have also another definition is the major allele or the minor allele. The major allele is called for the allele that have the major proportion in the population. For example, in this population here, we have 4 individuals, and we have the RNG is repeat 6 times. 2 in 2 individuals with homozygous and 2 individuals with the heterozygous. Meanwhile, the C allele is a minor allele because it repeats in only 2 heterozygous individuals. We have another definitions called the minor allele frequencies. Minor allele frequency is actually defined as the second most frequent allele value. Why is the most second allele value? So, minor allele frequency is used to define as the most frequent allele frequency. But recently, the minor allele frequency are using to calculate how informative genetic variation could be, and it defined on the second most variable frequency. Let's take an example. So if I have one locus with 2 alleles, which is small, the first one is take 70% or 0.7. Secondly is 37%. So in this case, the minor allele frequency will equal to this one. But this one is a minor allele frequency which is 0.3. But in another case, when we have a gene with 3 alleles, A star, and small a, we have the first one, the allele frequency is 60%, second one is 30%, and small a is 10%. In this case, we have the minor allele frequencies equal to, which one do you know? The second one, exactly. Second most frequent allele, which is 0.2. So why minor allele frequency is important? Well, to answer this, let's come back to our SPR set. So in last week we have discussed about the SDR loci. We know that the FBI, using the set of 16 SDR marker to investigate about paternity index and forensic science. Now, I have a question for you. This set was applied very successfully to perform sub-tests in US citizens. Now, if a Vietnamese scientist wants to bring this set to Vietnam and perform the same test for paternity testing and criminal investigation in Vietnam populations, is it possible to use the same set? You understand the question, right?
I think if we were to bring the set to Vietnam, wouldn't be possible for criminal tests because the SDR markers for the testing of criminal investigation was taken in the US population, and so the set of alleles and genes in the US population must be different than that in Vietnam. So to assume the test wouldn't be correct anymore.
So you think it was? So the first is impossible because it depends on the allen frequency. Do you, any of you have another answer, other explanations? Maybe other opinion? It's possible to apply the same set with the US to apply in Vietnamese populations. Anyone? Okay, so to answer this, please remember that in the bioinformatics practical assignment, you remember there's an exercise to estimate the allen frequency in different populations. You remember it? I know I remember exactly exercise 5 or 6, when they want you to estimate the allen frequency in different populations. So, the purpose of that exercise is to note that the allen frequency of each marker in different populations are different. And when we apply a test, we have to maintain that the accuracies of the set in a certain point. Let's say that we need to obtain the accuracy of the DNA test must more than 99.99%. That means that this DNA set couldn't wrong more than 0.01%. Right? So let's come back to our example. In here we have a frequency of marker, the first marker. So the father is 13, 15, the child is 13, 14, so the child is 13, 14 father. But the alien frequency of the 13 in the population is 0.308. That means 30.8% of the man out there, the alien frequency of this one out there in the population is 30.8%. Right? So, marker number 1, the alien frequency is in the population is 0.308, equivalent to 13.8%. Now the second marker, the alien frequency is 2738 and father 2732. So child must include the 27 from father. And the frequency of the 27 in the population is 4.1%, which is 0.041. Okay? That means only 4.1% of men out there have this marker. So if we combine of the two marker 1 and marker 2, we will have the ratio of the random men out there have the two marker 1 and 2 is 0.308 times 0.041, which is equal to, can anyone help me with calculations? 0.0126. 28. Okay. That means 1.3% of the men out there have the same two allele, 1 and 2. Right? So if we combine all the 16 markers, we will obtain a number, and this number must have to less than 0.01%. Here, we can have allowed the identical rate more than the identical rate of the 16 SPR marker more than 0.01%, corresponding to the accuracy of DNA test much more than 99.99%. Okay, so imagine that if one of the marker, for example this marker, the frequency in the population is 0%. That means all the population out there carry no allele 27. It's all carry the allele 32. So that means that this marker are not have a variable, cannot have any value in categorize one individual to the next. Right? So if one marker is too rare in the populations, that we consider that the frequency of the minor allele frequency close to 0, that means this marker have no value to identify, to apply in the forensic science and quantitative testing. Okay? For example, we are talking about this. So this is the distributions of this NIP. Let's say this is a marker in different populations in the world. So if we using this as a marker for testing, genetic testing, we see that in Vietnam, for example, 100% of the populations is G. We don't have anyone have A allele here. Okay? So if we're using this allele, all the population, no matter where he is, who he is, he will carry the genotype GG. You don't have to run the test to say that, oh, he's definitely GG because he's Vietnamese. Okay? And if in this case, the marker can have a value in categorized to calculate the accuracy of the populations, because the accuracy now is 100%. All of the big population carry the same gene. Okay, so this marker will have not have the value in genetic applications in Vietnam, in China, in Japan, in North Europe also, because in North Europe, in the vital circumstances, all the population is A. So we don't know who, we cannot use this value to categorize which individual to other. It's only have the value in the population that we have part of the population is G and part of the population is A. Like in India, in the East Africa, in the middle of America. Okay? You understand this point? Right. Okay, so that's the end of chapter 4. Do you have any questions? Now come to chapter 5. Mendel. The story life of Mendel is quite extraordinary. He was born in a very poor family that his parents cannot afford the luxury, expensive education. So he was sent to the church to become a priest. And as a priest, all his education fee was paid by the church. When he finished his career as a priest, he's come back to his hometown and start his career as a local teacher. He teach in math and physics. A couple of years later, he started in gardening in his church and start to study about the inheritance law of genetics. Do you know that actually Mendel was the first case in the history of science were catch after cheating in science? So the story is when Mendel doing his research on the pea plant, he hired a gardener to help him with the collecting pea plant, measure the physical trait, and quantify the obtained result. But we don't know that he did it in purpose or not. He told the gardener the expected result of each cross, each calculation. And the gardener, in the effort to keeping his job, throw away all the excess pea plant that not follow the 3-to-1 rule. So a couple of years later, actually a few decades later, mathematics Fisher, who invented the Fisher exact test famous, feel that the observed data of Mendel are too perfect. Even if the theory of Mendel was right, this result is too perfect to be true. He recalculate the result of Mendel and see that even if theory of Mendel was right, this result is nearly impossible to obtain in real life. So for example, if like you throw a coin 100 times, or you throw a dice 600,000 times, okay? A dice will have 6 possibilities, 1, 2, 3, 4, 5, and 6 points, right? If you throw 600,000 times, you will expect that 100,000 times you will have 1 point, 100,000 times you will have 2 points, 100,000 times you will have 3 points, and so on, right? But actually, in real life, it's impossible to obtain this perfect result you will have like 99,995 times, you will have 123 times, you will have 2.99,985 times, you will obtain 3 points, so on. So the result is too perfect to be true. And when Fisher calculate the possibility, even if the theory were right, is less than 0.0.0.1. That means it's too hardly to believe that this perfect number can happen in real life. And that's when the story was found out. Anyway, nowadays, the law of Mendel was proved right in a certain term and still valuable to study genetic until this day. So Mendel had invented 3 laws: the law of dominance, the law of segregation, and the law of independent assortment. Depending on the book, they will take the law of dominance is the first law or the third law. First, let's watch a small review on the Mendel law of dominance.
This study of genetics is incomplete without understanding the laws put forth by Mendel. After his experiments of monohybrid and dihybrid process, Mendel concluded that these are certain factors which are passed from one generation to the other. But he explained this better with the help of his first law in genetics. The first law in genetics is called the law of segregation. To understand this law, we first need to understand the concept of alleles. We know that genes, in simple terms, are the units of heredity that carry genetic information. Alleles are nothing but the different forms of genes. Let's take a simple example to understand this. Assume we have these 2 chromosomes. Here we have genes for the character height. Can you tell me which genes could possibly be present here? We can have either both capital TT or both lowercase tt. And we can even have one capital and one lowercase tt as the gene set. Now here, the individual T, that is the single gene, is nothing but the allele. So the alternative form, that is either dominant or recessive, is called allele. In simple words, one single T or T is an allele. Now that we know what alleles are, let's try to understand the law. There are different definitions you will find for this law, but let's pick the simplest one. It says that during gamete formation, the alleles for each gene segregate from each other such that each gamete formed carries only one allele for each gene. It may still sound a bit confusing. To understand this, let's take a simple example. Let's assume we have this single cell with only 2 chromosomes in it. The chromosomes have a set of genes, CC, for the skin color in an individual. Now to understand the law, let's break it into parts. The first part says, during gamete formation. This is the process of meiosis. It is the type of cell division which gives us 4 cells with half the number of chromosomes. The second part says the alleles of each gene. So here, C and C is what we're referring to. That means one allele from the gene set. Now what's the next part? The next part of the statement is segregate from each other. This means the alleles will separate. They will not remain together in the process. And now comes the last part. It says that one gamete carries only one allele for each gene. Now this part means that at the end of the meiosis process, we will get 4 cells such that each cell will have only one allele in it. So here, two cells will have capital C and the other two cells will have the lowercase c allele. Thus, each gamete gets only one allele. To summarize, we can say that the alleles for a gene segregate during gamete formation. To be honest, this law sounds very obvious to us now. It's like having a big book with many chapters in it, and then we make smaller books for each chapter. It is obvious that the smaller books will have the content resembling the content of the big book. This law sounds obvious also because we know the process of meiosis. But nobody knew this back in the earlier times. Hence, it was hypothesized by Mendel and then it was accepted as a law later. So what's the next law? It's the law of independent assortment. Does this mean that the genes assort independently? Or is there anything else that the law specifies? Let's find that out in the next part. There is one common thing among sea waves, dust particles, and a dice rolling on the floor. Any guesses what that common thing could be? It's randomness. Randomness is commonly found in nature and in several things around us. But can you state an example of randomness that occurs inside us? Though our body seems to be organized, there are a few processes that have no fixed pattern. One of the processes is that of the alignment of chromosomes on the metaphase plate. Let me elaborate. Imagine this to be a cell that's about to undergo division. For now, let's assume it's undergoing meiosis. We know that during cell division, a stage named metaphase is reached where the chromosomes align themselves on the equatorial plane. In other words, on the metaphase plate. So can you tell me how these chromosomes will align before they line up on the plate? Will all the red chromosomes from each pair line up on one side, or will they align in an alternate manner? Or maybe like this? Or will the alignment be like this? There are numerous ways in which this can happen, and this is random. There is no fixed pattern or a principle of sequence that the chromosomes follow when they align. Now, if the chromosomes get separated randomly, it's obvious that the genes present on them will also get separated randomly. So let's say this pair has the genes capital A and lowercase a. This pair has the gene set capital B and lowercase b, while this one has capital C and lowercase c on it. We are considering all the sets to be heterozygous in order to understand the concept better. So when the chromosomes get separated in the respective cells, it's obvious that the genes will assort independent of each other. There is no thumb rule that all the dominant alleles will perpetuate in one cell and all the recessive alleles in the other. They can assort independent of each other. This is exactly what Mendel had to say in his second law of genetics. It's named the law of independent assortment. The law says that genes for the different traits assort independent of each other during gamete formation. Let's understand this with the help of this cell. The first part says genes for different traits. This means we are addressing the genes which represent diverse traits. So here we have the gene sets AA, BB and CC on these respective chromosomes. These are the genes for different traits. Now the second part states assort independent of each other. This means the genes will not have a fixed pattern to follow when they segregate in the cells. So here, the alleles of the gene set AA will assort independent of the other two gene sets. It's not mandatory that all the dominant alleles will get assorted in the cells together. So the allele capital A can get assorted with any allele from the gene set BB and CC. And that's the reason why we get the gametes with different possible combinations at the end. To quickly review, the assortment of alleles is purely random and does not have a predictable and fixed pattern during the process of gamete formation. That is what the law says. There can be more possibilities of obtaining different combinations depending upon how the chromosomes align themselves on the metaphase plate. Now tell me, which type of a cross will help us understand this law better? A monohybrid cross or a dihybrid cross? Think about it. Let me help you with this. If it's a monohybrid cross, then there will be no independent assortment of the different alleles. And why is that so? That's because we are considering only one character at a time. So we will be dealing with a single gene set. And thus, this law is best understood when there are more than one character studied at a time. This is nothing but the dihybrid cross or higher crosses which involve more characters. Now that we're well versed with the first two laws of genetics, let's have a look at the third law in our upcoming part. A scene of lush green trees covered with yellow or brown mesh is not new to us. We have come across it many times. Any idea what this mesh is all about? These are also plants, parasitic plants to be precise. Being parasitic, they derive all the nutrients from the host and grow to form a huge structure. These plants are like creepers and overpower the huge trees as they grow. This is one of the best examples for understanding the fact that dominance is not a matter of size. Rather, it's never affected by size. It's the nature of the dominating component that matters. Let's take our genes for example. We know how small genes and their constituent alleles are. Yet, there is one form which dominates the other. We are familiar with the concept of dominant genes and recessive genes. But this wasn't known back then, not even to Gregor Mendel. That is the reason why he put forth the concept of dominant and recessive alleles with the help of this law. This law is called the law of dominance. After the monohybrid cross, Mendel concluded that a few genes are dominant while the others are recessive. Let's have a look at what the law has to say. The third law of genetics, known as the law of dominance, states that some alleles are dominant while others are recessive. An organism with at least one dominant allele displays the effect irrespective of the presence of the recessive one. The law is actually very simple. Let's take an example and understand it. Let's consider a cross of a tall and a dwarf plant. The first part of the law says that some alleles are dominant while others are recessive. Here, can you tell me why is this plant tall? That's because it has both the dominant alleles. We represent it with the capital letters TT. Similarly, this is a dwarf plant as it has both the recessive alleles denoted by TT in the lower case. Now the next part of the law states that an organism with at least one dominant allele displays the effect irrespective of the presence of the recessive one. So what does that mean? Here, on crossing the two, we get these plants in the F1 generation. The phenotype of all these plants is tall. And what is their genotype? As we can see, they are all heterozygous and with one dominant and one recessive allele. Doesn't the tall phenotype clearly indicate that the dominant allele has masked the presence of the recessive one? That's right. That's what the second part of a law says. The presence of a single dominant allele is enough to express the trait phenotypically. And what if both the genes are dominant? It's obvious that the phenotype will be dominant. The law states at least one dominant allele, which means if one or even both the alleles are dominant, then we get the respective dominant phenotype. Now let's cross this F1 generation to check whether we have understood the law correctly. The cross of these F1 hybrids gives us these offsprings in the F2 generation. Can you help me with the phenotype and the genotype of these offsprings? Here we get 3 tall plants and 1 dwarf plant. Genotypically, there is one plant that is homozygous for the dominant trait. There are 2 plants which are heterozygous, and this last one is again homozygous but for the recessive trait. So can we relate these results with our law of dominance? There are 2 types of alleles, one dominant and one recessive. This is what the first part says. And the second part says that at least one dominant allele displays the effect irrespective of the presence of the recessive one. So as we can see here that these 3 plants are tall because they contain the allele T for tall trait. This one has both the dominant alleles and these 2 have one allele for the dominant trait. Its present masks or suppresses the effect of the recessive allele. So this was about the third law of genetics called the law of dominance.
So the Mendel part you have all studied in high school, so I will go very briefly through this. So in Mendel, we choose to cross between two breeding parents. For example, in this case, we choose to study in two breeding with the phenotype is the color of the flower. So he defined that if the factor that the same between different was the same in one parent, that means the individual is in the homozygous genotype. And if the individual carry the 2 different alleles, that means they are in the heterozygous form. So when it's crossed with 2 true breeding parent with the phenotype in the rest is the color of the flower. The purple one crossed with the white one. Because they're 2 true breeding plants, so the genotype will be the same. They are in the homozygous state. So as a result, the F1 generation, because the F1 will be inherited, one gamete from the father, and let's consider this one is father, and the other one is from the mother. So we will, the F1 will have both alleles, big R and small r on the both parents. And why the F1 has the heterozygous phenotype? He will show one of the phenotype from one parent, and Mendel called this is dominant because while under the heterozygous form, it will still show the phenotype the same with the homozygous form of the parent. Okay? Which in here is purple color of the flower. Now the second law of Mendel, one is continue to cross the F1 to obtain F2. For example, when he studied about the color of the pot, like here, crossing 2 parents of the true breeding pig plant, one with a green color of the pot, and the second is at the yellow color of the pot. So in here, F1 is both heterozygous because it inherited one allele from the one parent and the other allele from the other parent. And follow the first rule and the rule of dominance. The green part is complete dominance against the yellow part. So all F1 will have the phenotype of the dominant trait, which is the green color. The second law, the Mendel start when he take the F1 and start the random mating inside F1 to auto-termination. So in F1, they will have the gamete is big G and small g with the ratio is 50% and 50%. Because also with F1, so the ratio of F2 is you have the genotype ratio is 1 is homozygous of big G, and 2 with the heterozygous with big G, small g, and 1 is homozygous of the small g, small g. And the ratio in the phenotype is 3 of the dominant phenotype, which is green part, and 1 is the recessive phenotype, which is the yellow part of the pea plant. And he has performed 7 traits in total in his career. The funniest thing today is sometimes there's a question why Mendel chose this 7 trait. Because this 7 trait right now have been proved to be located in different chromosomes. If there are 2 traits in the same chromosomes, that means they are linked. So the theory of Mendel can be applied. Because since the 2 traits are linked, they will have to follow the Markov rule, right? The rule of linkage disequilibrium. But we don't know that he chose accidentally or on purpose or not, but chooses different, 7 different traits located in different chromosomes. Some hypothesis that Mendel has studied many different phenotypes, and which phenotype follow his rule is given, and which phenotype are not following his rule is discarded. We don't know it is the truth yet. But nowadays, rule of Mendel still have a valuable, but we know that besides these 7 traits, there are 1,000 other traits that not following the Mendel rule, since they have located in the same chromosome, which we have to apply in the linkages. So the Mendel chose to study 7 different traits: color, flower, purple versus white, the position of the flowers in the middle of the plant or in the terminal on the top of the plant, the color of the seed, yellow versus green, the shape of the seed, brown versus wrinkled, the shape of the pot, inflated versus constricted, pot color, and the length of the plant. And all goes to the ratio 3 to 1. We have to figure out the second rule method, the rule of separation. And because 3 to 1 is 4, is combination of 2 and 2, which is having to figure out there are 2 factors of navicoid allele in each individual to determine a phenotype of interest. Now, the third rule of Mendel was invented when he tried to cross the 2 phenotype interests, which here is in a, like I will cross between the color of the pea plant and the shape of the pea plant. The yellow versus green and brown versus green. So when he crossed the 2 traits, you see that in the parent, the 2 group breeding yellow, 2 breeding parent, one will have a yellow and brown pea, and the other will have a green and wrinkled pea. So the F1 follows the first rule of Mendel, the rule of domination, also the dominant phenotype, which in here is yellow and brown, while they have a heterozygous genotype. And then, when the F1 self-pollinate, it will obtain the ratio of the F1 is heterozygous of yellow, positive yellow. Of the heterozygous, which is big Y, small y, positive Y, small y. Then the ratio of the S2 will have 3 yellow to 1 green. And the same with the color of the shape of the pea plant, which is big R, small r, positive big R, small r. We have 3 brown and 1 blue. So 4 combination time with 4 combinations, we will have total 16 combinations, in which yellow and brown is 3 times 3, which is 9, right? And green and brown we have 1 times 3, which is maybe 3. Yellow and winkle. And last one, 1 green and 1 winkle. 1 times 1 is 1. Green and winkle. 9 through 3 through 1. It makes 16 combinations. And here is the table of contribution between each gamete. So we have 4 type gamete, big Y, big R, big Y, small r, small y, big R, and small y, small r. Each will take 25%. You can check it 20%. So the 3 log methods based on the meiosis that we have discussed in chapter 3, when a gamete producing cell make a meiosis through 1 time the cell will double and 2 times the cell will divide, will produce the gamete with the number of chromosomes was divided by a half compared to the mother cell. And this distribution is happened totally randomly. So for example, in F1 generation, when we have both 2 paired chromosomes are heterozygous, it undergoes meiosis and providing gamete, we will have from 2 pairs of heterozygous genotype provide 4 types, 2 power by 2 which is 4. 4 types of gamete which is big R, big Y. The first combination here. Small r, small y. For second combination, big R, small y, and small r, big Y. Third combination here. So when we have the ratio of this, each one is 25%, we will have 4 types, which is power by 2 is 16 combinations leading to the ratio 9 to 3 to 3 to 1, and we can apply to 3 set of traits or 4 set of traits, 5 set of traits, and more. So now, let's reboot your mind with a small exercise. Please calculate for me in the course with genotype like this. Calculate for me the probability of the offspring with the dominant phenotype for the 4 traits. Please. Any answer? Yeah, please.
My answer is 3/16.
3/16. You all have the same answer, right? 3/16 is the correct answer. So in here, when we done the crosslink between the two, we will have the most parent is first one is big A, small a, big B, small b, big C, small b, cross with big A, small b, big C, small c, and big D, small b. Okay. Now let's analyze each pair. Okay. The first one, big A, small a, cross with big A, small. So in this case, we will have the gamete will be big A, small a, cross with big A, small a. So the genotype will be 1 big A, 2 big A, small a, and 1 small a. And the phenotype is 3 big A and 1 small a, right? So if they ask for the probability of the first dominant phenotype, we will have is in 3/4. Okay? The second one, big B, small b, cross with small b, small b. We will have the ratio here is 1 big B, and 1 small b. So the ratio is 1/2, 50%. The third one, big C, cross with big C, small c. So in this case, all the F1 will inherit at least 1 big C. So the percentage of having the phenotype is 100%. And in the last one, big D, small d, cross with small b, we will have the gamete and the ratio will be 1/2. So when we have combined all this, we will have the ratio of having the dominant for the both phenotype is 3/4 times 1/2 times 100% times 1/2, which is equal to 3/16. Okay, so now we're going to have a small break, 5-minute break, before we go further in our today's session. 5-minute break. Okay, so we have discussed about Mendel rule. So now let's come to the exceptions of the Mendel rules. So let's overcome all the parts related to gene linkage, which nowadays we don't know that Mendel chose the 7th trait just randomly, just by luck, or he did it on purpose and threw away the trait that not following his rule, the rule of independent assortment. Let's overcome also the natural selection, where one of the 2 traits was eliminated out of the populations based on the vitality of the carrier. The other term, factor can be related to the exceptional breakdown the rule of Mendel could be the mitochondria DNA, incomplete dominance, codominance, and we will discuss through this in the next part. First of all is about the mitochondria DNA. We have discussed with each other on the mitochondria DNA in chapter 1. We know that mtDNA is a circular DNA, double-strand DNA, circular DNA, and it's really stable. No cross-link, less ratio of having mutations, and quite stable from generation to generation. One thing is the mitochondria is mostly we inherited mitochondria from our mother part. Recent publications show that we inherited some mitochondria from our father, but not much, just a few mitochondria. So most of the mitochondria, let's say in one cell we have 4,000 to 5,000 more mitochondria. Most is inherited from our mother part. Different with the nuclear DNA, which the half of the DNA was inherited from the part and half was inherited from the mother part, and also there's a cross-link in the meiosis. So, come back to mitochondria. It's very easy to trace back the original from the mother part to the mitochondria, because most of the mitochondria was inherited exactly like the mother, since we inherited it from our mother and it's barely changed during inheritance. Right? So one group of science have applied this technology to trace back and also the original of the human race. Let's see what is. Let's see a claim about it.
Humans inherit mitochondrial DNA only from the mother. So in order to pass on mtDNA from generation to generation, the mother must have an unbroken link of female descendants. Unlike nuclear DNA, which undergoes recombination, much of mtDNA is passed from mother to daughter, relatively unchanged. If we take a sample of mtDNA from a population and try to reconstruct their matrilineal ancestry using the very slow mutation rate of mtDNA, we might arrive at a single female common ancestor, a mother whom we could all relate to from our mother's side. This is exactly what Kahn, Stoneking, and Wilson did in 1987, and found out that, in fact, all humans alive today do have a common female ancestor who probably lived about 200,000 years ago in Africa. They named her mitochondrial Eve. Analogous to this, tracing Y chromosomal DNA, which is only inherited from father to son, points to our common father, Y chromosomal Adam. However, there is no reason to believe that mitochondrial Eve and Y chromosomal Adam lived at the same time. Likewise, they were not the only humans living at the time, but their contemporaries failed to produce a direct matrilineal or patrilineal descent. Also, mitochondrial Eve and Y chromosomal Adam were only the most recent common ancestors. Going time, we can even find common ancestors of species all the way back to the first single-celled organisms. What's more, our actual common ancestor following both mother's and father's side probably lived as early as 5,000 to 10,000 years ago. All this shows that we are so closely related, more so than we could ever imagine.
Okay. Trong sinh học thì có một cái kỹ thuật gọi là đồng hồ phân tử. Khi mà các nhà khoa học tìm thấy một cái mẫu vật của một người cổ đại, người ta không biết là người ấy sống cách đây bao nhiêu nghìn năm, người ta sẽ tách cái đoạn DNA ti thể này. Vì DNA ti thể nó có như buổi đầu tiên là mình đã nói rồi, nó mặc dạng một vòng này, nó không có trao đổi chéo, nó rất là bền và rất ít khi xuất hiện genetic mutation. Cho nên người ta sẽ dựa trên cái tần số xuất hiện mutation, ví dụ trong khoảng độ 100 năm thì sẽ có một mutation, và người ta trace back lại cái hệ thi thể của người đấy so với con người hiện đại thì có bao nhiêu mutation, và người ta tính ra được là cái ông đấy sống cách đây bao nhiêu nghìn năm. Cùng với cả cái phương pháp gọi là đồng vị phóng xạ, từ đồng vị phóng xạ thì cũng có thể suy ngược lại xem là cái mẫu vật này đã có cách đây bao nhiêu nghìn năm, thì phương pháp đồng hồ phân tử cũng là một cái phương pháp như thế. Thế thì cái clip này nói về cái chuyện là một nhóm các nhà khoa học tại Viện Max Planck Đức khi mà nghiên cứu cái hệ gen ti thể của cả nhân loại thì thấy rằng là hệ gen ti thể của cả nhân loại có rất nhiều điểm chung, và cái đặc điểm là hệ gen ti thể là chỉ di truyền là dòng mẹ. Cho nên ông ấy dự đoán là khi mà có nhiều điểm chung như này thì trước đây đã có một cái này là học thuyết chưa chứng minh hoàn toàn nhé, đưa ra một cái học thuyết gọi là mitochondria Eve là người mẹ chung cả nhân loại, với cái giả thuyết là toàn bộ nhân loại trên trái đất hiện giờ 7 tỷ 8 tỷ người trước đây là có cùng chung một bà mẹ. Từ bà mẹ đấy sinh ra những người con đấy sinh ra toàn bộ trái đất này. Dĩ nhiên là điều đấy không có nghĩa là cùng thời mitochondria Eve sống là chỉ có một mình bà ấy là phụ nữ. Thời đấy có rất nhiều phụ nữ khác, nhưng mà những người phụ nữ đấy sinh con, sau đó con không truyền được đến thế hệ sau, đến một đời nào đó thì con vô sinh hoặc là không truyền được đến thế hệ sau chẳng hạn, thì cái bà mitochondria Eve này đã truyền được đến thế hệ sau. Và do đó là dựa trên cái dự đoán là cả nhân loại này đã có cùng chung một bà mẹ, và bà mẹ đấy sống cách đây khoảng độ 200.000 năm. Và cái học thuyết này cho đến giờ thì được công nhận rộng rãi, tuy nhiên là chưa chứng minh. Giống như là học thuyết Big Bang, đúng không? Big Bang của Stephen Hawking cho đến giờ là cũng chưa được chứng minh một cách hoàn toàn. Nên là một số học thuyết thì đã được chứng minh là sai rồi, ví dụ thuyết tiến hóa của Darwin hiện giờ chứng minh là sai rồi. So, a publication was published in Science in 2016 to explain why we inherited most of the mitochondria from our mother's side. Even in the sperm, there's some mitochondria too. So in the left image, you can see that the mitochondria in the sperm, which mark in the immunofluorescence in the green blood. However, when the sperm make a first contact with the egg for the fertilization process, the egg have a signal to synthesize an enzyme encoded by the SCPS6 enzyme, which is cleavage all the mitochondria from the sperm and keep only the mitochondria from the egg. However, sometimes this process can have an error, and leading consequences is sometimes we have the mitochondria from our father too. The mitochondria is encoding for most of the energy synthesized in the cell, and the error, the mutation in the human genome can lead into certain disorders, mostly related to the intellectual development, but also related to the other diseases.
Mitochondrial diseases cover a wide range of conditions caused by genetic mutations. In the UK alone, they affect thousands. Mitochondria are the powerhouses of the cell. They use a process called oxidative phosphorylation to generate energy. But a mitochondrion is a complex piece of machinery, and a lot can go wrong. It takes around 1,500 proteins for mitochondria to function, and problems with many of these can lead to disease. The cell's nuclear DNA creates all but 13 of these proteins. The rest are coded for by the DNA in the mitochondria themselves, and mutations in either can cause problems. A cell has just one nucleus and many mitochondria. All a cell's mitochondria contain their own DNA. If a mutation affects mitochondrial DNA, some might be healthy, others defective. And different cells can have different numbers of defective mitochondria inside them. That means that some cells may be less affected than others. The diseases caused by damaged mitochondria can also be varied. They can affect any organ or tissue, and they can appear at different times throughout life. For example, Leigh syndrome is the most common type to develop in childhood. It can affect patients in a number of ways, including loss of skills, epilepsy, and problems with muscle function. Mutations in over 75 genes in both nuclear and mitochondrial DNA can lead to this syndrome. So what can be done to tackle mitochondrial diseases, like Leigh syndrome? Treating patients is difficult, but in some cases the disease can be stopped before it's passed on. Mitochondria are inherited from the mother. If the mutation is in the mitochondrial DNA, this inheritance can be blocked. One approach is to combine the nuclear DNA from an affected egg cell with healthy mitochondria from another woman. The embryo would then have nuclear DNA from the original mother and father, and mitochondrial DNA from the egg cell donor. This has led some to call such potential infants three-parent babies. This technique is still very new, but it has recently been approved in the UK. It may be a controversial approach, but techniques like these could finally help combat these devastating diseases.
Pre-parent technique. So the pre-parent technique was actually is quite new. And the first was applied in UK. This technique is to cure a disorder caused by mtDNA mutations. So in this case, the mother whose mtDNA genome was mutated, carrying disease, will be the egg of the mother will be take the nucleus out and combine with the nucleus from the sperm from the husband, and put the two inside of the other egg of the donor woman. The donor egg will be remove the nucleus and put inside the zygote of the combination of the mother and the father, and the babies was born with the inherited material from three different individuals. Half of the nuclear genome is from the mother, whose mitochondrial DNA was mutated. Another half is coming from the father, who's healthy, and the mitochondrial genome is come from the donor woman, who provide also the cytoplasm for the eggs. So he or she actually have three parents, and literally. And so this technique was first applied in the UK, and here is Dr. John. He is holding the baby. Was the first baby was born with three-parent technique. Thực ra thì trong một cái hội thảo mới đây thì các nhà khoa học cũng đã công bố luôn là cái kỹ thuật ba bố mẹ này còn được áp dụng khi mà các cặp đồng tính nam muốn sinh con. Các cặp đồng tính nữ muốn sinh con thì trước đây đã công bố rồi là hoàn toàn có thể khi mà người ta lấy cái nhân của một người phụ nữ và combine với cái nhân của người phụ nữ khác và giữ nguyên cái vỏ trứng của người phụ nữ đó. Thế còn cái three-parent technique này là khi hai tinh trùng lấy nhân của cả hai tinh trùng ra và lấy một cái tế bào trứng của một người phụ nữ hiến cái trứng đó và loại bỏ cái nhân đó đi và cho cái hợp tử hình thành bởi hai tinh trùng vào bên trong, và dĩ nhiên là vẫn phải cấy vào tử cung của người phụ nữ đó để mang thai hộ. Nhưng mà cái kỹ thuật này thì invent là kỹ thuật tương đối mới và là tương đối thế hệ. Alright, so the next exception of the Mendel inheritance is the incomplete dominance. So in here, the incomplete dominance, that means the dominant allele is not completely cover the phenotype of the recessive allele. For example, in the flower here, we have the parent who homozygous with the red color and white color. And in F1 generation, we have the individuals in F1 who have the heterozygous genotype, and they will have the intermediate phenotype between the red and the white. These have the pink color here. So if the F1 undergo self-pollinated, we will obtain the F2 with the ratio is 1 to 2 to 1, exactly like the genotype ratio. 1 to 2 to 1. Another example of the Mendel inheritance is the codominance. For example, like in the blood group. Okay? In blood group, we have 3 alleles. This trait was controlled by 3 alleles. The IA and the IB is codominant, and the small I is recessive. So small I is encoding for O blood group. One in the red blood cell, in the membrane of the red blood cell, there is non-antigens, and in the blood plasma there is antibody against A, both A and B. In the blood group A, is controlled by the dominant allele IA, so the genotype could be both IA or IAI, small I. So in this case, the red blood cell is covered by the antigen A, and in the antibody in the plasma is anti against B. In the B blood group, we have the red blood cell was covered by the antigen B, and the antibody is against A. And in AB group, in the red blood group, we have both the antigen A and B, and in the plasma, we have none of the antibody. So if the antibody against A and getting to the blood group red blood group A and get the antigens A, it will have immunoreactions and make a blood clot. So it will stop the bloodstream and prevent the bloodstream to carry oxygen and carbon dioxide in and out of the body. One thing is funny about the blood group. Since the blood group O have many type antibody in the plasma, and the AB blood group have not antibody in the plasma, a group of scientists have performed analysis to see which blood group show the strongest immune response against infectious disease and also cancer. And they see that which blood group have more type of antibody in the plasma. For example, like blood group O is have a better immune response against the infectious disease and better reaction against cancer cell compared to the other. And in the other hand, the other blood group which have non-type is antibody in the blood plasma, so the most susceptible against the infectious risk and also cancer cell. This have a very interesting result, interesting information related to blood group. The next exceptional Mendel rule is polygenic trait, which right now is a nightmare for any geneticist. Polygenic trait is that the phenotype and trait were not controlled by one locus, but controlled by several locus genes. In that case, scientists have to scan for the genome and based on the method called positional cloning to see which region of the gene have the DNA fragment related to the disorder. Let's say most of the disorder that been fully studied right now is a single gene trait or single gene phenotype. The polygenic trait or polygenic disorder right now is still considered as a black box, dark material in some cases. And finally is epitasis. Epitasis is a phenomenon where the express of locus was controlled by another locus located in the different chromosomes. For example, in a lab rat here, which is a tiger dog, the trait of the color of the fur, the pigment, was controlled by one pair of allele in the locus called big B and small b. The big B is the dominant and encoding for the color black one is produce the black pigment for the fur, and the small b is encoding for the brown, that is encoding for the color for the brown for the fur. However, this pair allele was taken control by another pair, which is the pair E here. If the big E's were exists in the genome of the carrier, then the dark pigment can be synthesized and the dog can have the color black or brown. In the other hand, if the genotype of the other locus is small e, then the carrier cannot have the ability to synthesize the dark pigment. Thus, they will have the color of the fur is yellow as follows. So in here, the genetic pigment was controlled literally by one pair allele, but also this pair allele was also controlled by another allele in the different chromosomes. So in general, we see that phenotype is controlled by two pair genomes, two pair alleles. Okay, the next part related to human disorder, I think we can save it for the next section. So do you have any questions for today's sessions? Any section, any confusions? Okay, so you have no questions, thank you very much, and see you